So welcome to the second installment of carbohydrate physiology and associated metabolic processes with an emphasis on transport mechanism across cell membrane. The key topics covered in this part 2 includes number 1, the composition and function of the lipid bilayer within the cell membrane, 2, various cellular mechanism responsible for moving substance across this membrane. And lastly, a detailed examination of how glucose is transported through membrane using facilitated protein carrier, which are glucose transporters. Now, let's start with plasma membrane. Okay, the plasma membrane is a flexible, meaning not rigid, as you can see in the animation, contains phospholipid bilayer that separate the cell's interior, which is the intracellular fluid, and from its environment or the extracellular fluid. Its phospholipids have hydrophilic heads and hydrophobic tails, which is represent the fatty acids, creating a fluid amphipathic structure essential for selective permeability. This allows small non-polar molecules to pass freely, while larger or large molecules require specific transport proteins. Now, the role of cholesterol. Cholesterol molecules within the membrane help maintain its stability across temperatures. Now, the membrane proteins are also present and either in the integral or embedded or in the peripheral surface attached. The integral protein serves as a channel or receptors, and even glycoprotein involved in the cell recognition and communication, including the glycocalyx, a sticky layer aiding binding and signaling. Now, in terms of fluidity of the cell membrane, it is influenced by whether the fatty acid chains in the phospholipids are saturated or unsaturated. As you can see, in the saturated fatty acid have straight chains on your left that fit closely together, resulting in a stiffer membrane. Conversely, unsaturated fatty acids have bends caused by double bonds which hinder tight packing and enhance membrane flexibility. Now, this interplay allows cells to adjust membrane characteristics according to temperature and functional demands. Now, beyond fatty acid composition, there are several other factors that may affect plasma membrane fluidity, as outlined in this table. This includes temperature, pH change, organic solvents, and protein content, along with the aspect we've already covered, that's fatty acid saturation, phospholipid tail length, and cholesterol. Now, cholesterol presence in the plasma membrane, since cholesterol play a vital role in preserving the fluidity and stability of the plasma membrane by balancing its rigidity and flexibility. It enables cells to adjust to environmental fluctuations such as temperature shifts, mechanical forces, and chemical stresses, while regulating membrane permeability, facilitating the formation of lipid rafts for signaling and supporting overall cell's function and responsiveness. In response to environmental temperature changes, cholesterol decreases plasma membrane fluidity at high temperature to prevent leakage, while low temperature it increases fluidity to avoid membrane stiffness. Now, cells use various transport methods to move substances across their membrane, We have simple diffusion, facilitated diffusion, and osmosis are all passive processes, meaning they move molecules down their concentration gradient from high to low without requiring energy or ATP. Especially, osmosis refers to the movement of water towards areas with a higher solute concentration. Now, simple diffusion involves small nonpolar molecules like oxygen and carbon dioxide that can easily cross the plasma membrane. In contrast, facilitated diffusion also moves substances down their concentration gradient but involve larger molecules that cannot freely pass through the membrane, so they require a protein carrier such as glucose transporter, which we will explore in detail later. The direct active um, transport uses ATP directly to move substances across the membrane, typically against their concentration gradient, like the sodium potassium pump, while in the indirect active transport, on the other hand, relies on the energy stored in the ion gradient, not ATP directly, to drive the movement of other molecules, often through symport and antiport mechanism. Both are essential for maintaining cellular homeostasis and enabling nutrient uptake. The last one is the bulk transport. Involves energy dependent processes where endocytosis brings substances into the cell and exocytosis expels them out. Examples include phagocytosis for endocytosis and hormone release such as insulin secretion for exocytosis. Now, this is the schematic diagram of a passive diffusion, facilitated diffusion, as well as active transport with the use of energy, ATP. So, in summary, facilitated diffusion is the major glucose transport method, especially in insulin sensitive tissue. But active transport also play a key role in the absorption and reabsorption for especially SGLT1 in small intestine for glucose absorption, and both SGLT1 and 2 in renal tubules glucose reabsorption. Now, this is the sodium potassium pump, an active transport, primary active transport. It is widely regulated by scientists as one of the most vital active transport mechanisms in the body. The sodium potassium pump plays a key role in maintaining the cell's internal environment by managing ion balance. It is essential for establishing the resting membrane potentials, facilitating nerve and muscle function, regulating cell volume, and generating ion gradients that drive other transport processes. This pump is fundamental for normal cellular operation and overall physiological health. Now, when we look at the difference between active transport and facilitated transport, the active transport can involve co-transporter mechanism on your left known as symport or antiport. In symport, two molecules move together in the same direction across the membrane, while the antiport, the molecules move in opposite direction. Both these processes rely on energy stored in ion gradient rather than direct ATP use. In contrast, on your right, facilitated transport typically functions as uniport, where a single substance moves across the membrane independently. The perfect example here is glucose transport through a glucose transporter. Now, it's important to discuss secondary active transport with the use of SGLT1 and SGLT2. Now, SGLT1 and 2 are sodium glucose transporter that use secondary active transport to move glucose into the epithelial cell by coupling it with sodium ion. The sodium ions moves down its concentration gradient. That's on the left side of the slides, the lumen of the proximal tubules or the lumen of the small intestine. again, the sodium moves down its concentration gradient, pulling glucose against its gradient into the cell via SYMPORT. Now, this process does not directly use ATP at the transporter. Now, to sustain this mechanism, the cell must contain or maintain a low intracellular sodium concentration. This is achieved by the sodium ATP pump on the basolateral membrane, which uses ATP to actively pump sodium out of the cell and potassium into the cell, preserving the sodium gradient that powers both SGLT1 and 2. Once inside the cell, glucose exits into the bloodstream through GLUT2 facilitated diffusion transporter on the basolateral side, that's on your right side. GLUT2 allows glucose to move down its concentration gradient into the circulation without requiring energy. And this process complements the secondary active transport. So that concludes my presentation of part 2. And in part 3, we will shift our focus to glucose transporters, the GLUTs, and the isoenzymes, glucokinase, and the difference between glucokinase and hexokinase. So thank you for listening.